Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the immunoglobulin G1. From the last couple of videos, we are talking about the isotypes or the classes of the antibodies, and we are discussing on the immunoglobulin G and the different subtypes of the immunoglobulin G. And we are currently focusing on the immunoglobulin G1. In the last video, I've told you about the important immunological functions that the immunoglobulin G1 can play, like this is a strong activator of the complement system, the classical complement system, and it has a very strong antibody dependent cellular cytotoxic activity. Now, in this particular video, I want to focus on the placental transfer of the immunoglobulin G1. And this placental transfer of the immunoglobulin G1 is very important to provide passive immunity from the mother into the fetus. Is the immune system of the fetus is still developing, it have got a weak immune system, so the immunoglobulin G1, when they are transferred from the mother into the fetus, these IgG1 are providing passive immunity to the fetus. Now, how this particular transfer is achieved? If you look at the structure of the placenta, there are specialized cells which are known as the trophoblast, and a subclass of the trophoblast, they are known as the Syncytiotrophoblast. And these particular cells, they are very important to mediate the transfer of the immunoglobulin G1 from the mother into the fetus. Now, how does this work? If you look at the term Syncytiotrophoblast, that is actually a Greek word, and the syn mean together. The cytio actually refer to cells. The tropho mean nutrition, and blast mean bird. So this Syncytiotrophoblast is actually the epithelial covering of the embryonic placental villi, which invade the wall of the uterus to establish nutrient circulation between the mother and the embryo. So that would mean that it is very important for the nutrition, but at the same time, this is very important for the placental transfer of the immunoglobulin G1. Now, how does this particular mechanism work? If you look at this one, this is the maternal blood. You've got physiological pH around 7.4, and there are immunoglobulin G along with some other serum proteins. So during the process of the endocytosis, you are going to have an acidified endosome. You can say this is the cells of the Syncytiotrophoblast. We are discussing the mechanism in these particular cells. So once they endocytosed, the acidified endosomes, you have got specialized receptors which are known as the FcRn, or which are the neonatal Fc receptors. So you are going to see these neonatal Fc receptors on the Syncytiotrophoblast. What these neonatal Fc receptor do, that they are going to bind to the immunoglobulin G1. The immunoglobulin G1 are using their Fc region to interact with the neonatal Fc receptors, and once they are bind, this binding is actually facilitated by the acidic pH around 6. And so this particular acidic pH is going to promote the interaction of the immunoglobulin G1 with the neonatal Fc receptor. Once in the acidified endosome, the neonatal Fc receptor has bound to the immunoglobulin G1, then you are going to go for the sorting, because when you are going for the endocytosis, some other serum protein, they are also coming along, but we are not interested in those particular serum protein. So we are going for the sorting of the neonatal Fc receptor and the immunoglobulin G1 complexes, and we are going to remove the other non-receptor bound proteins which are actually degraded in the lysosomes. So this particular complex of the neonatal Fc receptor and the immunoglobulin G1, when they move towards the fetal side of the placenta, the process of the exocytosis occurs, and when you go towards the fetal side of the placenta, the pH becomes around 7.4. Just like the maternal blood, there is physiological pH. So on the fetal side, you are going to see this physiological pH around pH 7.4. Now, this particular physiological pH is not going to support the interaction of the neonatal Fc receptor with the immunoglobulin gene, or you can say that the interaction between the immunoglobulin G1 and the neonatal Fc receptor that is very weak around the physiological pH. So the immunoglobulin G1, they are going to get released into the fetal blood, where they can provide passive immunity to the fetus. We will continue our discussion on the other subclasses of the immunoglobulin G in the next video.